I work in imaging, mainly magnetic resonance imaging, and we've been working for some time now, maybe the last 20-25 years, on developing imaging methods to detect very early evidence of treatment response, so that you could use that then to guide treatment. And obviously I'm going to talk about imaging brain tumours, mainly PDX models of brain tumours. And we've been developing a new technique, a new metabolic imaging technique with MR, that massively increases sensitivity and allows us to interrogate the metabolism of the tumour essentially in real time. And we can use that method to detect very early evidence of treatment response. We see very early changes in metabolic activity post-treatment. So it's a way of massively increasing the sensitivity of magnetic resonance. Magnetic resonance is an intrinsically insensitive technique because you're looking at very weak interaction between the nucleus spin and the magnetic field. And this technique works by, you can imagine some atomic nuclei behave like small bar magnets. And some of them line up with the field, but some of them actually, because such a weak interaction, line up against the field. But it's that population difference. You have slightly more spins aligned with the field, and it's that population difference which gives rise to signal. So the physics of this goes back to the 1950s, in fact. But a group working in Malmö in Sweden back in the 1990s developed a way, well, as I said, it had been worked on for many years, but they made a practical way of polarizing the spins, making them all line up. And you do this at very, very low temperature. But they developed a way of rapidly warming the sample back up to room temperature and retain that spin polarization. So transiently, you have this massive gain in sensitivity. You then take this material that you've polarized the spins and inject it intravenously, and this has been done in patients now. And for a few minutes, you have a huge amount of signal that you can then, so you can track where this labeled molecule goes in the body. But more importantly, you can see its metabolic interconversion with other metabolites. So it's sort of real-time biochemistry. You can watch in real time how this molecule was taken up by a tumor cell and subsequently metabolized. And that process, we know changes with some drugs, have a very direct metabolic effect. So you see almost instantly a change in the way that they behave. It takes, say, instantly 12, 24 hours, 48 hours. I mean, it takes a while, but you nevertheless see these changes. So in fact, the discussion was this morning about pharmacokinetics and pharmacodynamics. So essentially what we can tell you in some instances with some drugs that the drug actually has engaged with its target. It's hit the target. You know that from this imaging experiment. So for example, people talking about labeling drugs and looking at drug transport, well, that may or may not tell you whether it's actually got into the cell, for example. But a pharmacodynamic readout tells you the drug crossed the blood-brain barrier, it's got into the cell, and it's hit its target as well. So we see this as a way of a sort of image-treat paradigm where as more and more drugs become available and drugs targeted at particular types of tumours, or we know that not everybody responds in the same way. When you've got a whole family of drugs or a spectrum of drugs that you could use, or you might want to use drug combinations as well. So I think if you can image very quickly whether the drug has worked or not, then that's of benefit. You can clearly, if a drug isn't working, you can switch to an alternative drug or an alternative protocol or administration protocol, for example. So that's, I see it as complementary to other methods that already be. So for example, there's been a lot of discussion about sequencing, and you can choose drugs on the basis of known mutations in a particular tumor. Of course, that's very valuable, and you can choose the drug up front, but we think you could then follow up with imaging to see whether that drug is indeed working or not. There's over 20 instruments now been sold worldwide, and in the UK, there's 4 sites now, 4 or 5 sites that have got the instrumentation. Some activity in cardiac, but mainly in oncology. So it's still a very early stage of development. Sort of proof of concept, we know it will work in patients, it's gone into the clinic. But now what we really need to understand, can we do something useful with this? Is it really going to have some major, is it going to change clinical practice? And clearly that's what it needs to do to get more wider adoption. And I think we're at that stage where we've shown in principle it works. We can get signal and we see changes in the signal. The next stage is in wider clinical studies, can we use this now in a very productive way clinically? Yes, I mean there are, particularly as you might imagine, in the US, but there are also sites in Europe as well which are working with this technology. But although it's now 15 years since the first practical demonstration of the technique, it took about another 10 years to get first into clinic. I think now the hard work, I think, is really going to begin, because we need, and of course at this stage it's quite expensive to do these studies as well, but we need to look at lots more patients and different diseases as well. I mean, on the basis of preclinical data, we think we know where it might work, but we need to demonstrate that now in patients. Oh, I think it's still watch this space. We're very excited by it, to actually see it go into the clinic for the first time, but we also recognise it's going to take a while before we really understand what we can do with it.